Good morning. I am Dr. Pankaj Kumar, Associate Professor of Medicine. I'll give a talk on hemolytic anemia. Now, hemolytic anemia is a form of anemia where hemolysis occurs. That is, there is abnormal. Normally, also RBCs are broken down in the reticular endothelial system of the spleen after 120 days. But here, the breakdown of RBCs is abnormal. And what is the abnormality? That abnormality is that either they are broken too early, that is, they are broken before 120 days, and secondly is that hemolysis can be of two types. One is the intravascular hemolysis, that is, the RBC, the breakdown takes place inside the blood vessels, or there can be extravascular RBCs, hemolysis, where the hemolysis takes place not in the blood vessels but in the reticular endothelial system of the body. Now, if we talk about the classification of hemolytic anemias, the problem can lie inside the RBC, which are called the intracorpuscular defects, or the problem can lie outside the RBC. If we talk about intracorpuscular defects, the problem can be inherited or acquired. Acquired, the example is paroxysmal nocturnal hemoglobinuria because it has to do with a PIGA gene, which is an autosomal somatic mutation. So it is not taken as an inherited mutation. Somatic mutation is not genetically transmitted. So it involves mutation of the PIGA gene, which is a somatic mutation. But inherited defects are also there, and these inherited defects can be in the membrane, the cytoskeleton of the membrane of the RBC, which can be to abnormal shapes like pseudocytosis, maybe erythrocytosis. The problem can be with the enzymes. Two important enzymes are the pyruvate kinase and the G6PD, that is glucose 6 phosphate dehydrogenase. Or the problem can lie in the hemoglobin, that the hemoglobin is abnormal, which can again be of two types. It can be a qualitative defect or a quantitative defect. The qualitative defect is sickle cell anemia, and the quantitative defects are the various kinds of thalassemias. Now, as far as the extracorpuscular defects are thought, there can be familial problems leading to hemolytic uremic syndrome, but there are a lot of mechanical causes or a lot of causes which are required and can cause extracorpuscular hemolysis. And these are, there can be microangiopathic hemolytic anemia, which we have already discussed. Then there can be toxic agents like Clostridium, snake venom, etc. There can be drugs which can lead to hemolysis. There can be infections, and on the top, the infection is malaria, which can lead to hemolysis, or there may be autoimmune causes. Now, hemolytic anemia can be acute or chronic. The first thing to be understood when we talk about the clinical features of hemolytic anemia are whether the hemolysis occurred acutely or whether it occurred in a chronic manner. We must have seen that there are patients who are having thalassemia who have got a hemoglobin of as low as 6, 7, but they are not that symptomatic. But if there is acute hemolysis, then it can cause severe problems. So chronic hemolytic anemia are well tolerated. They are not that poorly tolerated, but acute hemolysis, as for example in the case of G6PD deficiency or some crisis-like situations, can lead to severe problems. This is akin to these two cities. One is a village in Meghalaya, Mao syndrome, and it is the place where there is the highest rainfall. But as you can see, people are comfortably walking around because Though it is a chronic severe problem, but it is over a period of time so well tolerated. So goes for chronic anemias, even though the problem is there, but it is well tolerated. On the other hand, there is a desert city of Jaipur, where even a few centimeters more than the expected rainfall can suddenly produce devastating floods. Now, effect of hemolysis is that the bone marrow is stimulated to produce and increase its output by 6 to 8 folds. 6 to 8 folds. The red blood cells are released. In the process, a lot of premature reticulocytes and reticulocytes with polykinesia are also increased. Now, once the supply is and the balance it is impaired, that means the destruction is more and person is not able to synthesize that many RBCs, the anemia results. Effects of free hemoglobin in the blood. Now, when we were talking that hemolysis can be intravascular or extravascular, one of the main concerns is that the hemolysis, if it is intravascular, is dangerous. If it is extravascular, it is less dangerous. Why so? Now, in intravascular hemolysis, the hemoglobin comes freely into the blood. It starts circulating in the blood as hemoglobin. Normally, hemoglobin is never found in the serum. It is always found inside the RBCs. So, it now comes into the urine and starts appearing in the urine as hemoglobinuria. Please remember, hemoglobinuria and hematuria are not the same thing. Hematuria means passage of intact RBCs. Hemoglobinuria means passage of only hemoglobin. So this is one thing. This hemoglobin contains iron in the ferrous form, which can damage the cells through the Fenton's reaction, which we will just see. Now, other thing is, so it can lead to shutdown of the kidneys. But on the other hand, if the hemoglobin is normally like it is destroyed inside the reticuloendothelial cells that are macrophages of the spleen, then the heme is properly tackled and it is not allowed to be released. The ferrous iron is not allowed to be released. It is converted into ferric iron and then it is bound to proteins like ferritin or transferrin and then only it is released in the blood or stored. So it doesn't cause harm from that angle. Of course it will cause anemia, but at least it will not cause the toxic effects of circulating hemoglobin which are due to the ferrous state iron inside that. This is a diagram depicting the Fenton's reaction when the ferrous iron of the heme moiety reacts with hydrogen peroxide. It produces ferric iron, but it also produces a hydroxyl radical, and this can damage the tissues. Second thing is that the free hemoglobin binds to nitric oxide. Nitric oxide is synthesized by the endothelial cells of the vascular system. Very short half-life is there. So, but nitric oxide is very important because it causes vasodilation. And if nitric oxide is consumed, it will lead to various complications like pulmonary hypertension because of vasoconstriction, or maybe priapism, or intestinal spasm, or sometimes other smooth muscle spasms can be there because of the consumption of nitric oxide by the circulating hemoglobin, and the patient may have dysphagia, etc. Okay, this is a diagram of the nitric oxide. It is synthesized. Nitric oxide is a messenger which causes the formation of cyclic GMP from ATP. That is what results in vasodilation. So consumption of nitric oxide by the circulating hemoglobin is bound to lead to vasoconstriction. same thing. So, if we now try to see the consequences of hemolytic anemia, there is first of all anemia. Second thing is there is rise in the unconjugated bilirubin, which causes jaundice, because once the hemoglobin is broken, it is dissolved into globule, which is harmless, it is thrown into the blood, but the heme has to be converted into biliverdin, and then it is converted into bilirubin. So bilirubin goes to the liver, but if the production of bilirubin is increased, it will lead to unconjugated hyperbilirubinemia. Then increase in urinary urobilinogen, increased reabsorption of urobilin from the gut. Increased LDH is an important consequence of hemolysis, because LDH, which is an enzyme present in the red blood cells, is released. Reticulocytosis, we have discussed that they are a reaction to the decreased hemoglobin, and the bone marrows may increase the production of the RBCs by 6 to 8 fold. So reticulocyte counts are also released in the blood in that proportion only, and sometimes a little bit more are released. Folate deficiency, when so much of synthesis is going on of the RBC, 6 to 8 folds which are increased, so it requires substrate. So substrate problems can occur, especially the one which is required is the folate, and sometimes folate crisis can occur because of this. These are the physical consequences of hemolytic anemia. These are all because of the extramedullary hematopoiesis. The girl on the right hand side is typically having a pinched nose, malar prominences, bottom of the skull. This is indicative of the extramedullary hematopoiesis. The boy on the left hand side also indicates a lemonish stain because of unconjugated hyperbilirubinemia. This is a crew-cut appearance, and the pinched nose of the boy is seen. Now, if we talk about the overall complications and consequences of hemolytic anemia, of course, what we have discussed will be there, but there will be also an important thing will be there, that we will have to give transfusions repeatedly for hemolytic anemia, which will result in transfusion overload, iron overload. When repeated transfusions are given, it results in iron overload. In fact, you have to give chelation therapy when you are giving too many blood transfusions. The iron overload itself can have many manifestations. In the brain, it can attack the neurohypophysis and the pituitary stalk, and this can lead to diabetes insipidus. Then it can affect the heart and can lead to cardiomyopathy. Then it can affect the joints and can lead to arthropathy related to iron overload. It can cause pancreatitis. It can also lead to destruction of beta cells, which can result in diabetes. At the same time, it may cause a pigmentation in the skin, and the combination of skin pigmentation and diabetes is called Crohn's diabetes. It may lead to testicular atrophy. It may lead to organomegaly. So there are many consequences of iron overload because of repeated transfusion, and because of the anemia also, as we have seen, typical facies or something can be there. So these are again revising that how the hemolytic anemia will manifest depends upon whether it is acute or chronic. If it is an acute process, the process will be very severe and it will not be tolerated well. But if it is a chronic process, the body makes arrangement and it is able to tolerate it well. Second thing is the site of hemolysis is very important, because if it is intravascular hemolysis, it is more dangerous. It can lead to consumption of nitric oxide, it can lead to Fenton reaction, and variety of thrombi can occur. It can pass on into the hemoglobin can go into the urine, can cause hemoglobinuria, and can even shut down the kidney. A small diagram showing the same thing, that either the RBCs are extravascularly destroyed in the reticular endothelial system or they are destroyed inside the circulation. Now, when the RBCs are destroyed intravascularly, there is a protein which is called haptoglobin. Haptoglobin is supposed to form a complex with the free hemoglobin and thereby protect the body. So once the haptoglobin is consumed fully, then of course the hemoglobin will start to appear in the urine. Now, so the consequences of this are that there will be decreased haptoglobin and there will be increased methemoglobin, which means that it has got a iron in the ferric state in the hemoglobin. So this will be there, and this methemoglobin will bind to the albumin and it will form another complex which is called methylmalbumin. Basically it means that hemoglobin is there in the blood circulation itself. Then another protein which is called hemopexin, that will be consumed, and then there can be hemosideruria. This is because there is hemoglobinuria, some amount of this hemoglobin which is being excreted is taken up by pinocytosis by the cells of the urinary tract, and later on these cells are shed, causing the production of hemosideruria. Role of spleen in hemolysis. Now, spleen is the main place where the senescent, worn out RBCs, they are removed from the circulation. A few words about ferritin. It is a universal intracellular protein that stores iron and releases it in a controlled fashion. But hemosiderin is a particle resembling iron storage complex that is formed by breakdown of hemoglobin or abnormal metabolic pathway of ferritin. So hemosiderin is also there. Only thing is that hemosiderin does not release iron in time of use very quickly, but ferritin releases iron when required a little bit more energy. Hemosideruria. In hemoglobinuria due to chronic intravascular hemolysis, the hemoglobin is absorbed by the proximal tubular cells where the hemoglobin iron is converted to hemosiderin. When these tubular cells are later shed in the urine, it results in hemosideruria. The excess of bilirubin, which is formed because the substrate, that is unconjugated bilirubin, is increased, can lead to certain stones in the gallbladder, which are called the pigment stones. Chronic hemolytic anemia increases the level of unconjugated bilirubin in the blood and biliary tract. Unconjugated bilirubin is insoluble in water and can precipitate with calcium salts or stones in the presence of stasis or gallbladder infection. You see, normally the gallbladder, it excretes mainly the conjugated bilirubin, but when unconjugated bilirubin is in large amounts, it can appear in the bile and it has the capacity to cause the stones, especially if there is an nidus of infection. This is a normal appearing RBC. We have already seen in our previous slides. It has got 1/3 of the area is pale, 1/3 of the diameter is pale, and it's a biconcave disc. So these are the other various sites. Types of RBC. There can be a normal, then there can be a macrocyte more than 100 microliters, a microcytic hypoclinic cell. The size will be less than 80 femtoliters. Elliptocyte is a elliptical cell. Target cell is where the central area of panel is occupied by some color pigmentation because of the shape of the cell. Teardrop cell, sickle cell can be there. Acanthocyte means protections are there for the cell, and spherocyte means a large cell, not a large cell, a cell with no central area of color. So spherocytes are small dark cells which suggest autoimmune hemolysis or hereditary spherocytosis. Sickle cells, they indicate a hemoglobinopathy. And red cell fragments, or they are also called schistocytes, are a sign to account for microangiopathic hemolytic anemia. These are the spherical cells, spherocytes. They have no area of central pallor. Sickle cells are shaped like sickles. They occur in hypoxic conditions. These are the red cell fragments or the schistocytes.